So we come to what is the final topic, perhaps the most important in this discussion on sequencing at a genomic level. And this module has to do with an introduction to you to next generation sequencing. Next generation sequencing is literally the state of the art today. The genome of an entire species can be determined. Say, I look for a job in bioinformatics. Then you realize that bioinformatics is often synonymous with what is the state of the art. When we were looking at the definition of bioinformatics, I indicated several modules ago that bioinformatics was often synonymous with what was going on at that time, sequence analysis, for example. So when you thought of bioinformatics, you really associated it with all the work that was being done related to sequence analysis. It's the same thing that's going on right now, and next generation sequencing um, produces a lot of data, and bioinformatics methodologies and new methodologies, software, etc., are used to process this data. And that's why, now if you really look in terms of what is bioinformatics called, and it's called big data. And that has to do with the results, the whatever products of next generation sequencing. The experimental part is this data, and the processing of the data requires an incredible influx of bioinformatics effort. Right now, you would say uh, high throughput methodologies is where the bulk of the bioinformatics effort is going towards. So, for those of you who are interested in pursuing this further, there is a course conducted by the European Bioinformatics Institute, and this is an online next generation sequencing practical course. It is the course is several modules, each of them ranging from 10 minutes or so to more than an hour. And if you're really interested in doing this, I would certainly recommend that you take this course. Sequencing is determining the composition of DNA or protein sequence in the correct order. That is, what is the order of the nucleotides in a DNA, or the nucleotides in an RNA, or the amino acid order in a protein. NGS is also very often referred to as massively parallel sequencing because of the need to do uh, many sequences essentially simultaneously, and then kind of stitch them into the sequence of a chromosome, and then by extension the sequence of a genome. So Sanger sequencing was used to determine the order of a nucleotide sequence one at a time. And now we can go take a look at this video. We've already have had a discussion on Sanger sequencing in next generation. So but we will look at this video anyway.
Welcome again, friends, and this particular video will be talking about the Sanger method of DNA sequencing. Now, DNA sequencing method developed by Nobel laureate Frederick Sanger was the method used to sequence the genomes for Homo sapiens and a variety of other organisms. Now, Homo sapiens, remind, this is us, right? Human being. In these huge projects, the target DNA from human, fish, dog, etc. is broken up into small pieces and packed into small loops of bacterial DNA known as plasmid. As the bacterial cells grow and divide, they replicate the plasmid, which is here act as a target. Now this target DNA will be replicated, as well as their own DNA is replicated in the bacterial cells. In preparation of sequencing, the plasmid containing the target DNA must be separated from the rest of the bacterial cells. Robots and detergent to break open the bacterial cell and release the DNA. These samples are placed in centrifuge to spin. This process gathers the heavy components of the cell at the bottom of the tube. Only the plasmid DNA carrying out the target sequence is left in the solution. This plasmid DNA is filtered and washed to further purify the sample. The solution containing the plasmid with the target DNA sequence is transferred to another type of plates. All of these things nowadays are done by robotics. And the chemicals required to start the sequencing reaction are also added during this time. The sequencing mixture of plasmid and target DNA, DNA nucleotides, and fluorescently labeled or modified nucleotides, enzymes and primer sequences to get the reaction started is then put onto the heating blocks. So remember what are put? Plasmid DNA, nucleotide sequences that are required. Here is the DNTPs because deoxyribonucleotide sequences are needed. Then the primers and also enzymes are provided, and some fluorescently labeled modified nucleotides are also supplied. Let us see how these ingredients combine in the Sanger reaction. The reaction is initiated at either of the end of the target DNA, whether it joins with the plasmid DNA. It it's easier to see the action if we show the DNA unwound, right? So let us consider the DNA at its unwounding state. First, the DNA is heated so that its two DNA strands start to separate. And this is done by increasing the temperature to 96 degrees Celsius. The temperature is then lowered so that a short length of the DNA sequence, which is called a primer, that is complementary to the known plasmid DNA sequence, can bind to it. Now for this, we need to lower down the temperature to 50 degrees Celsius. Finally, the temperature is raised slightly to enable the enzyme to bind to the short section of the double-stranded DNA. Now the enzyme that starts to make a new strand of the DNA by incorporating free nucleotides that are complementary to the target DNA. Now for this we must slightly raise the temperature from 50 degrees Celsius to 60 to 65 degrees Celsius. The enzyme will continue to extend the strands until it randomly incorporates a fluorescently labeled nucleotide. The fluorescently labeled nucleotides are chemically altered so that they terminate the DNA sequence or they terminate the DNA strand synthesis and the enzyme falls away. The cycle of heating and cooling is repeated many times, generating a large number of fragments of different lengths that end in fluorescently labeled bases. For example, here it is end at this point. In previous case, it was extended at this point. Now in this case also it will be extended into some other point. So what we are getting here, we are getting the different DNA segments with different variable lengths. And at the end of each DNA segments here, single stranded DNA segment, we are having a fluorescently labeled altered base. So it is going on and different types of populations are produced. At this stage, the fragments are floating freely in the many of the tiny tubes in a plate known as wells. To sort the fragments out by size and detect which bases have been added when, the plate is loaded into the sequencing machine. Inside the machine, the samples are transferred into the glass capillaries, where an electrical charge starts to negatively charge the DNA molecules through a gel matrix. As they move, the longer DNA fragments are slowed down more by the gel, and the shorter fragments will start to migrate further in this run. So when all the DNA fragments from your sequencing reaction are run through the capillaries, they are sorted by the size from the shortest to the largest. Shortest one is going to be the fastest one, largest one are going to be the slowest one. And the laser at the end of the capillaries is used to excite the final fluorescent basis, which is recorded as a colored peak or a bar. So each time when it passes through the cell sorter, it will measure the fluorescence effectivity and it will give a peak or a bar. This process means that the fragments that incorporated fluorescent tags and terminated early will be read first. The laser can detect only the fluorescently labeled bases at the end of each fragment. So remember, at the end of each fragment, the presence of the fluorescent bases are very, very important, and it's a key for the understanding of this Sanger sequencing concept. So each colored bar represents the final base of each strands of the DNA that was made in the sequencing reaction ordered from shortest to the longest. Capillary sequences can be used for many samples at a time. This output shows 96 samples or rows and can read the equivalent of 500 to 800 DNA letters from each sample. Now, this method has been used by scientists around the world to decode the DNA. From its inception in 1977 through the early days of studying single genes to the Human Genome Project and beyond, the Sanger method has undefined our understanding of gene and genetics in health and diseases.
Okay, so again, I hope that was a nice little refresher on Sanger sequencing. In next generation sequencing, millions of fragments that arise out of a single sequence can be sequenced parallelly and then put together within a few hours to a day. Okay. Genome-wide sequencing for relatively smaller species is therefore possible in a very relatively speaking short time. Now, when I keep saying relative and relatively speaking, it depends upon the kind of computational resources you have. It's depending upon, if you have a faster computer, if you have gigabits of speed associated with your software, if you are associated with a high performance computing center like we have at Texas Tech. Um, that kind of support certainly mitigates the issues related to processing speeds. The issues of storage, however, that's a whole different issue. Okay, so the question is, why high throughput? So information began becoming available at genomic levels, right? So imagine the effort that would take to deal with one sequence, and we looked at that in terms of how you would, break up a sequence of interest in very strategic ways, and then use chromatograph and use chromatography methods in order to identify what those fragments are. Alternatively, you can use Sanger sequencing to build your sequencing based on a complementary the availability of a complementary sequence, and then you build it up, and along the building process using the deoxynucleotides you get fragments, and then you put stitch these fragments together into the sequence of interest. Okay. um, it was no longer feasible to work one nucleotide at a time. Now imagine one little sequence consisting of 20 base pairs, 20 nucleotides, and then say, well, what kind of processing would you need to do 3 billion base pairs, right? Which is essentially the length of the human genome. The researchers were seeking a faster and cheaper alternative, and this is where next generation sequencing methodologies came about. So, what are the advantages of next generation sequencing? As this discussion progresses, you understand that they're kind of obvious, but let's enumerate them anyway. It's a cheaper alternative than Sanger sequencing in the sense that, more importantly, that you can do several Sanger sequencing steps simultaneously, several hundreds, several thousands. Small genomes can be sequenced in a single day, so speed, the high throughput nature of it, a rapid discovery of genes and regulatory elements associated with disease. Targeted sequencing allows the identification of disease causing mutations for diagnosis of disease. So everything, and we discussed genome browsers and identification of SNPs and the potential for identifying SNPs associated with uh, matched, SNPs potentially matched with certain diseases. Now imagine if you could do all that really rapidly, identify all of these really rapidly, and that's where the advantages of next generation sequencing come in. And then you have a kind of a side method which also involves next generation sequencing, which is RNA-seq, and it involves the sequencing of the transcriptome, which is the mRNA of a species. Okay, the entire, like a genome is the entire genetic all the chromosomes, the nucleotide, essentially all the base pairs associated with the genome, and if you take a look at the mRNA of it, which is the transcribed part of the transcribable parts of the genome, is then called the transcriptome. Okay, and this information comes from nature. There are some disadvantages. Everybody doesn't have next generation sequencing equipment. Everybody does not have access to high performance computing. Very expensive. The costs are prohibitive. The initial step setup costs approximately $100,000. Small genomes cost approximately $1,000 to run. The costs are decreasing with time, but it's essentially the setup, right? The genomics facility run by Professor uh, Rao Kotapalli um, actually has some of that, and it costs a lot of money. You cannot really avoid errors in sequencing. They are present. Again, depends upon the nature of the processing, etc. These errors are reducing. Data analysis and visualization can be a major problem, and data storage is becoming a major challenge because of the large amounts. And you will see why even a single next generation sequencing experiment results in such a huge burden as far as storage is concerned. So let's look at the schematic. So you have template preparation. So you have a genomic DNA or a cDNA. Remember, we talked about mRNA being reverse transcribed using reverse transcriptase into cDNA. We've looked at that. And so you have your DNA. And then you have what is a step called as library preparation, where you take your DNA and you fragment it. And then you use a process of adapter ligation. And then you amplify um, the DNA, these fragments, and these fragments can be amplified onto, for example, microbeads or channels. Now, the Illumina high sequencing, the high-seq that we have actually uses these channels, and then you amplify onto microbeads or in channels, and then you use this process of bridge amplification that we will discuss. And once you have the bridge amplification, then you have large signals associated with these fragments associated with the gene. And then for example, you can use this methodology called iron torrent, or you can use this process called MySequence, which uses fluorescence to identify sequences. And once these sequences are identified, and you get what are called reads, which are these fragments that you've generated. Now these have to be stitched together into the large sequence of interest. Okay, and then it goes to data